Question?
Yes.
Yes?
Hold on. Oh, yeah. On the study guide, like, it has a couple of, like, specific antibiotics, and it says, talk about the general structure, the uses, and the mode of action. What do you mean by uses? Like, on what type of bacteria?
Like, just how far does it go?
What do you mean?
How far do you
Like, do we need to mention a couple things it treats, or?
Yeah.
So, like, gram-positive bacteria.
Um,
Gram-negative.
Or like TV or walking around?
yeah, things like TB, walking. Well, the examples that I gave in the lecture. Use the lecture notes that I post as kind of a guide for level of depth you've got to go into, because the book goes into more depth. But the lecture notes kind of give you, if you have that level of understanding, you're good. Yes, Audrey.
Can you explain the GALC? Is that
That's an acronym for gut-associated lymphoid tissue. And it's a subset of MALT, which is mucosal-associated lymphoid tissue. So that's kind of the generic term for lymphoid tissue associated with mucosal membranes. And the GALT is the stuff in the gut. Okay.
For the people in the chemotherapy study guide, do we just need to know like what they worked on, like specifically, not really like the whole, just what they worked on?
The whole story, you mean? We don't have that many people, do we?
I just don't like stories.
You don't like stories?
That's all I can do sometimes.
Do you think we had an exam coming up?
Yeah, maybe. 2 weeks. Wait.
Lab exam week
Yeah,
after.
there's a lot of them next week.
Yeah.
And a PChem exam next week.
Yes.
Which of the ID batch I do, one of them is due next week.
One ID Bacter is due by the lab exam. The rest are due by the final.
Okay. The mandatory.
One mandatory ID Bacter is due by next week for the lab exam. The optional are due by the final exam.
There's a question in here that says, is it normal for microbes to colonize new body fluids and organs? Yes. It says-
Wait. Body fluids and organs, no. Those are usually azenic, which means germ-free.
What has to happen in order for this to occur?
Yeah, they have to be able to penetrate the barriers and then multiply in those, and that is infection. Study guide questions. I like that.
I'm in the day.
Wednesday's lab actually does not take the whole period of time. And so Wednesday would be a good time if you have last minute questions too, to clean up some of those. So all we have to do Wednesday is we have to inoculate this thing called an enterotube. And then you're going to work on a little project which involves making a flow diagram, which will help to organize your thoughts for identifying your unknown.
Are we going to do the urea?
Yes, in fact, so yes, new urease auger works. Okay. So I'll have those out for you, and you can inoculate things on Wednesday. Anything else?
What factors influence the composition of normal flora?
What factors?
Environmental?
Yeah, foods you eat and things like that, but the factor age, sex, hormones, antibiotics. Okay, remember what we're doing right now. We're talking about the overall structure of the immune system, and we divided it into compartments. And we very quickly breezed through 3 of those compartments, really, and that is the extracellular fluid, the RES, and a little bit more depth on the lymphatic system. And now we're delving into this 4th compartment, the blood. So we talked about hemopoiesis. Remember what that means? Yeah, blood cell development, generation of blood cell populations, differentiation of stem cells into the erythrocytes, platelets, leukocytes. And we've been looking at that leukocyte arm of development and breaking it down into 2 major categories, which are the granulocytes and the agranulocytes. Approximate number of agranulocytes in your body, what percentage of the population are agranulocytes? 2/3. Okay. And about 1/3 are the agranulocytes. Did I say agranulocytes?
Yeah,
Sorry, granulocytes are 2/3.
2/3
Agranulocytes, 1/3. Okay. And we just started talking about the agranulocytes, and the first ones I mentioned at the end of the period were the monocytes/macrophages. Monocytes are the precursors of macrophages. When a stem cell differentiates and starts releasing monocytes into the blood, as long as they're in the blood, they're called monocytes, but they don't stay there very long.
Are they still functional in the blood, or are they not functional?
They're not functional in terms of what those cells normally end up doing, no. So they're kind of not fully differentiated, okay? But once they migrate out of the blood into the tissues, then they complete their differentiation into macrophages. Do you remember what part of that differentiation involves? There's a big physical change in those cells. They get a whole lot bigger. Okay, that's part of the differentiation process. They swell 5 to 10 times in size. So what do those macrophages do? What's some of their function in immunity? Remember, they're white blood cells now. Okay, phagocytosis is something that they're going to be involved in. What other cells have we encountered already that have phagocytic activity that are not agranulocytes or granulocytes? Neutrophils, good. Okay. Remember, the neutrophils are kind of the first ones to the site of infection. I liken them to kind of the frontline foot soldiers. Macrophages are more kind of the officers. You know, an officer in battle will kill if they have to, but they have a lot of other important roles as well. Macrophages, when they phagocytize a foreign body, they not only kill it and break it down, but then they display pieces of that broken down substance on the surface of their cell. And that's going to serve as a signal to other immune cells. That's called antigen processing and presentation. And we're going to get into that in more detail on the next exam, which, by the way, remember, the final exam is just the 4th exam. It is not a cumulative exam over the whole course of the semester. Because the immunology, yes, you can thank me now. The immunology that we're going to cover is in-depth enough that that's plenty to handle for a final exam.
Is that on Tuesday?
Yes, I believe it is. Tuesday at 8.
Wow.
So this is one of the important things that the macrophages will do that the neutrophils will not do. Okay? Another important thing that they do is they're involved in chemical communication with other cells, and particularly other cells of the immune system. And these molecules that they produce are called lymphokines. So secretion of lymphokines is another important thing to bear in mind. Now, one of the things we're not quite ready to get in yet is how does a neutrophil, or how does a macrophage for that matter, know that something is foreign and shouldn't be there, and that they should act upon it.
Has receptors on it, or
It's going to have some receptors on it. We'll talk about those receptors. They're called toll-like receptors. And we'll talk a little bit more about those, but we'll wait until we specifically kind of go through the steps of phagocytosis. Okay. Now, I mentioned earlier, and I can't remember what context it was, but the fact, when we were talking about the RES system, that macrophages oftentimes are subdivided into two populations, the fixed macrophages and the wandering ones. So the wandering ones are the ones that kind of just patrol throughout your body, checking things out. That group grope that I referred to, checking out the surface of the molecules of other things they encounter. Receptor contact's going to be really important. The ones that are, though, localized specifically in tissues are called the fixed macrophages. And here's a couple of examples of some fixed macrophages. This particular one is called an alveolar macrophage. And here's an alveoli cell. Here's a macrophage right here. And they patrol around within the alveoli looking for those inhaled, probably invading microorganisms. There are populations in the liver as well. Those are called the Kupffer cells. And then to make things confusing, there is this cousin to the macrophage called a dendritic cell. For practical purposes, you can really consider dendritic cells and macrophages to kind of be the same thing. They do many of the same things, okay? They engulf, they process and present antigens, they secrete lymphocytes. But if you look at this slide right here, which shows you the differentiation fates of various cells during hemopoiesis, what you're going to see is that here's the cell differentiation line for the granulocytes. And then I also mentioned mast cells, the ones that are similar to the basophils, but instead of being mobile, these are fixed within connective tissue. But then here, if we look at the monocyte differentiation, we've got a little bit different pathway for the dendritic cells than we do the macrophages. And then here's the differentiation pathway for the lymphocytes. Then we've got this other weird guy we'll talk about over here called the natural killer cells. So they're a little bit different in origin and probably slightly different in function. But these are some of the major players in immunity. And a lot of the stuff we talk about in regard to antigen presentation and processing really does happen with these dendritic cells. Okay?
So you had talked about how like these have receptors on them, and I say like no things are foreign, but can certain viruses have like receptors that specifically like target these cells?
Well, that would be a little counterproductive. Well,
Because like I don't
can these cells be infected by virus? Is that what you're asking?
I guess so. Because like
Yes, they can.
like with Ebola, like it says that it mainly like initially infected these cells, and those are its main target cells, the macrophages,
Yes. Yes, there are a whole variety of receptors we'll talk about next exam on the surface of these immune cells, and some virus that evolved the ability to bind to those receptors and infect those immune cells. Yep, absolutely. So here is an example of a dendritic cell, also called a Langerhans cell. Paul Langerhans was the guy who discovered, if you like, these dendritic cells. But one of the things you can see on these dendritic cells, and I think I mentioned it earlier on, is that their cell membrane has these long projections. I think Hannah asked if they were related to dendrites. Dendrites have the same kind of long projections on those neuron cells. So these are the Langerhans cells. In old books, these used to be called macrophages. Now we're understanding a little bit more sophisticatedly that they're actually dendritic cells that have slightly different origin during hemopoiesis. but functionally they behave pretty much the same way as macrophages do. So we'll come back and talk about phagocytosis and some of the toll-like receptors and things. Okay, the other big bucket of cells within that agranulocyte population are the lymphocytes. And the lymphocytes are really, really important in that third line of defense. The adaptive immunity. And we're really not going to get into these now except to make sure you understand their existence and generally what they're involved in until the last exam material when we'll talk about the lymphocyte response in depth. What were the two types of lymphocytes, remember? B and T, and remember that they have different places where they are educated. You remember what education involves?
The specific antigen that they target.
Yep, learning to recognize a particular antigen, and that happens because each cell is able to assemble a particular receptor that is complementary to a particular antigen. So education involves building your unique receptor. Okay? So with T lymphocytes, where does that happen? Thymus, B lymphocytes, bone marrow. Okay. And then what happened to those educated cells? Where did they go? They move out into what we call the secondary or regional lymphatic tissue. It might be the spleen, it might be the tonsils, it might be the malt, it might be the galt. Very likely will be the lymph nodes. And then they sit there in an inactive state until what happens? They encounter their partner antigen. And remember, those encounters usually take place within those lymphoid tissues. So if the antigen started out infecting skin cells, how does that knowledge that the infection was occurring get transmitted to the lymphatic system? That's where those macrophage dendritic cells come in. Okay, remember they engulf, ingest, present, process, and then migrate their way to this lymphatic tissue, where they then have these, B and T lymphocytes that have been sitting there their whole life waiting to do their job, and then finally they're kicked into action. When B and T lymphocytes are activated, two subpopulations of cells are produced. And they are called effector cells and memory cells. And remember the role of memory cells. Memory cells basically tell the immune system, you have seen this before. And if you see this antigen again, you will then be able to respond much more rapidly to that particular infectious agent. Effector cells do something in the actual immune response. So for B cells, the major effector cell is the plasma cell. And you remember what the plasma cell does? It's the main thing B lymphocytes do. They're involved in antibody production. You start cranking out that antibody. There are actually 3 effector cells in T lymphocyte populations. And those are the helper cells. What do helper cells do? They assist the B cells in making antibody. There are also another subpopulation of T effector cells called the cytotoxic cells, or sometimes also called the killer cells. What do they do? What does a T killer cell do?
They kill those cells that are infected.
They kill infected or cancerous host cells. Okay? And they do so in kind of an interesting way. They use kind of an interesting system called the perforin granzyme system. What do you suppose a perforin does? Punches holes in the cell wall that the T cell attaches to, okay? Or the membrane, sorry, because these are human cells. And then granzymes are these enzymes that enter through that rupture, and they ultimately stimulate the cell to go through apoptosis, which is programmed cell death. The final subpopulation of the T effector cells are the suppressor cells. And what do you suppose they do? What might they suppress? The immune response in general, okay? Because remember, we would prefer that diseases and the response against those diseases be acute. And once the threat is eliminated, do we want this immune response to continue? No. We don't want to have a chronic immune response. We want to have this resolved. And so the T suppressors help to shut down the immune response when the threat is eliminated. Now, because these B and T cells are part of the third line of defense, they have to be activated by specific antigens. So these are antigen-activated responses.
Do the suppressors like kill the other cells or not?
No, what they probably do is they do a couple of things. They shut down cells like the macrophages. They inhibit them from sending their chemical communications to other cells. And then the suppressors might produce their own chemical inhibitory signals. Okay? So, I mean, that's a good point, is they really kind of suppress the cells. You don't want to kill them because then you're compromising your ability to respond in the future.
I have another one.
Yes?
Why do the memory cells like last so long, and why can't we?
We'll have to get into that on the next exam.
Okay.
Yes. We're just not quite ready to be there yet. Okay, one of the other kind of cousins that I want to mention is this guy over here on the right. These are called the natural killer cells. They're another agranulocyte. But unlike the T and B cells, they are not antigen activated. You don't need a particular antigen to stimulate these cells. So these really constitute a fairly early response against infection. The B and T cells take a little bit of time to get ramped up. But these natural killer cells can start working pretty quickly. Like the T cytotoxic cell, they kill infected and cancerous host cells. And they do that by secreting chemicals on that cell surface. But they don't have to be activated by a specific antigen to do that. How do they get activated? They're activated by chemical stimuli, by lymphokines. And many of those lymphokines are produced during the process of inflammation. And we'll talk about inflammation probably tomorrow. But inflammation is part of your second line of defense. It's not part of your first line of defense. Okay, so those are the major players now that are going to be involved in the immune response. Now let's take a look at a couple of the interesting things that white blood cells can typically do. Some kind of unique properties of white blood cells. Now, not all of the cells have these properties, but most of the important ones do. One important property that some of the white blood cells have is the ability to do diapetesis. Or if you prefer a more technical term, leukocyte extravasation. Diapetesis is probably easier to remember. This basically is just a fancy way to describe the ability of these white blood cells to move out of the circulatory system into the surrounding tissues. And that can be a challenge because the endothelial cells that line the inside of a capillary, for example, have reasonably tight junctions. And that's important because if the junctions were really loose, what would happen? A lot of fluid would leak out of your capillaries into the surrounding tissues. So you'd swell up. But when you are infected in your tissues, let's say you get a scratch, you want those white blood cells, particularly the phagocytic ones, to get out there. So at that particular point in time, some changes do occur which loosen up these intercellular connections and make the capillaries more leaky. So my fingers represent intercellular junctions between endothelial cells. This is kind of the normal situation, okay? And it's very difficult for leukocytes to migrate out of that particular capillary into the surrounding tissues. But if those cells dilate, that tends to spread apart the junctions and stretch them a little bit. And then the white blood cells can extravasate or crawl out of the capillary and into the surrounding tissues. And that involves some interesting interactions between some cellular membrane proteins on the surface of the white blood cells and some membrane receptors on the surface of the endothelial cells. I think I mentioned earlier, that's what that inner life of the cell video that you probably have seen illustrates. It's that whole process of the white blood cell doing this diapetesis.
So the white blood cell stimulates the endothelial cells to swell?
No, typically what happens is the inflammation will release chemicals that cause that to happen. Then the white blood cells take advantage of it. Okay? Now, typically a part of this also is that the white blood cells are being attracted to the site of infection by a chemical attraction, chemotaxis. So they're moving toward a stimuli, in this particular case, a chemical stimuli, so we call it chemotaxis, and they're being attracted to that particular stimuli, so it's a positive chemotactic response. Where do you suppose some of those chemotactic factors come from? What would attract white blood cells? Pathogens. It might be molecules that are associated with the pathogens. It could be things that are produced by dead cells. Okay? Or there are also things that are produced during the process of inflammation. Another important thing that some white blood cells do, particularly the neutrophils and the monocytes, or the macrophages and dendritic cells, is they undergo the process of phagocytosis. And for convenience purposes, we typically will break phagocytosis down. Here's a white blood cell in yellow up here, phagocytosing a bacillus-shaped bacteria. And eventually that will result in destruction of that bacterial cell. Typically, we break phagocytosis down into various identifiable stages. Okay, so this slide kind of summarizes some of the stages of phagocytosis. Typically, the first thing that's involved in phagocytosis is that attraction to chemotaxis. And that draws the white blood cells to the site of infection. But really, what has to happen next that's really important is there has to be an attachment. Okay, the phagocytic cell has got to bind to the pathogen. If it doesn't, nothing else will happen. So first step is chemotaxis. And then attachment. Attachment really requires a matching or a complementarity of molecules on the surface of the pathogen and molecules on the surface of the phagocytic cell. And if you get the right attachment, if you get the right docking, if you like, then you can proceed on to the next step. What are the receptors on the surface of the phagocytic cell called? Well, there's probably more receptors than we understand. But one set of receptors that is pretty well characterized is a set of receptors called toll-like receptors, or TLRs. Right now, there are 10 recognized toll-like receptors. And this just gives you the number. They're usually numbered in order of discovery. And also gives you an idea of what they are looking for. What kind of thing on the surface of the pathogen are they looking for? These toll-like receptors bind to a group of molecules called PAMPs. PAMP stands for pathogen, so these are going to be on the pathogen associated molecular patterns. Ideally, these would be molecules you would typically find on the surface of a pathogen, but would typically not be found on the surface of a host cell. So for example, do our host cells have an LPS layer? No. So lipid A, which is an important part of the LPS layer, could be a PAMP. Toll-like receptors would then recognize a PAMP of lipid A, whether it was on an E. coli or whether it was on a Salmonella. It could respond to a whole group of pathogens as long as they had that particular molecular pattern. Okay? So it's kind of a generalized response to kind of, wow, if you have this, you must be pathogenic. Okay? So you see some things up here that constitute PAMs, okay? Lipopeptides, lipoteichoic acid. That's one of the things you find in the cell wall, gram-positive cell wall. Lipid A. Flagellin, that's a good one, okay, because our cells don't have flagellin. Even our flagellated cells have a different protein that makes up the flagella of eukaryotic cells. Some of those are found in the cytoplasmic membrane of the phagocytic cell, so on the external cell membrane. Some of them are actually found in the phagosome membrane, okay? If attachment occurs, what that stimulates is the process called receptor-mediated endocytosis, okay? So here's a receptor on the host cell. It binds to a pathogen. And when those two match up, the cell membrane invaginates or pinches in and eventually we form a vacuum. That's called a phagosome. Some of these toll-like receptors are actually located in the phagosome membrane. And they can respond to things like RNAs in viruses, single-stranded RNA, double-stranded RNA. Now, additionally, in addition to toll-like receptors, there are some receptors in the cytoplasm of the cell called NODs. Basically, go ahead.
Can I ask you, can you remind me what a phagosome is and what's the difference?
A phagosome is a vacuole, basically, that results when the receptor binds to the pathogen, so the toll-like receptor binds to the pathogen. That will stimulate this receptor-mediated endocytosis, resulting in whatever was engulfed and bound to those receptors being in the vacuole.
Okay.
So that vacuole is the phagosome.
Okay. So it's
Okay.
just another.
That's another name for vacuole. Yep. Okay. This just shows, I never showed this, this shows the toll-like receptor on the macrophage, and then here's the foreign molecule binding to that toll-like receptor. And when that happens, it not only stimulates phagocytosis, but the binding of the foreign molecule to the toll-like receptor causes this macrophage now to start producing some lymphokines, some signaling molecules. Okay? And some of those are involved in stimulating inflammation. So remember that these macrophages had a couple of roles. They phagocytized, they secreted lymphokines, chemical communicators, and then eventually, as this is engulfed, will eventually then on some cells display those antigens on the surface as well. If it's neutrophil, that won't happen. But if it's macrophage, it will.
Is there ever an instance where like a cell will bind with the antigen but never do anything with it, just kind of like
Yep.
take it up and just kind of die off?
Yep, we'll talk about that too. Yep. Okay, these nods are kind of like toll-like receptors, but instead they're in the cytoplasm of the phagocytic cell. So even if something gets into the cell and isn't digested completely, there can still be receptors in the cytoplasm that recognize it, and when they're activated, then they can produce some of these signaling molecules that will help to deal with the infection. So you've got both kind of an external and an internal set of receptors that are looking for things that are foreign. They have made some interesting discoveries about mutations in genes that code for some of these NOD proteins. And they found a really high correlation between mutations in these NOD genes and a pretty nasty condition called inflammatory bowel disease. Any of you ever heard of Crohn's disease before? Crohn's disease is a type of inflammatory bowel disease. And there's a high correlation between mutations in these NOD receptors in the cytoplasm of your phagocytic cells and that particular disease. Okay, so what happens next? Okay, we've got this phagosome produced here now. Typically, the next step is that the phagosome fuses with the lysosome. So now we have a phagolysosome. So what does that accomplish? What's in the lysosome?
The lysosome will start digesting the bacteria.
Yeah, there's all kinds of lipases and proteases. Typically, they're bunched together and called acid hydrolases, DNase, RNase. Okay, anything that breaks down big molecules into their monomers. Why are they called acid hydrolases? They have acidic pH optimum. They really only function in acidic environments. Is the lysosome acidic? Actually, it is. One of the things that the lysosome does is there are proton pumps in the membrane of the lysosome, and they pump hydrogen ions inside. So the lysosomal enzymes are nice and active. Why might that be advantageous to have your enzymes packaged into cytoplasm in this little mini acidic environment, and that's really where they have to be to work?
Could it have to do with the bacteria being deactivated or being affected by the acid?
Could be, yeah. That could be an additional killing mechanism.
So they want to be active elsewhere.
They don't want to be active elsewhere, correct. Okay, do you want those acid hydrolases to spill out into your cytoplasm and start digesting your proteins and things? No. Is your normal cytoplasm of your cell acidic? No, it's pretty neutral. Okay, so if the lysosomes do break, which sometimes happen, then when these acid hydrolases get out into the basic cytoplasm, what do?
Very good.
They denature, right? They unfold. They don't work properly. There's another set of killing mechanisms that are activated within the lysosome, and those are the oxygen-dependent killing mechanisms. One of the things that typically happens when a phagocytic cell gobbles something up is there will be something initiated called a respiratory burst. And the rate of oxygen utilization of these cells just skyrockets. More oxygen is being utilized, the more toxic byproducts of oxygen metabolism that are being produced. So those superoxide anions and things like that start being cranked out purposely inside of these phagolysosomes, and that really contributes tremendously to killing the pathogen as well. Okay. Additionally, there's lysozyme that's present in the phagolysosome. Remember what lysozyme does? Remember tears, saliva? Yeah. Tends to break down peptidoglycans, so it's very important in gram-positive infections. Okay. So what's the next step now? You've killed your pathogen and you've kind of busted it up. Then the final step typically is elimination And that phagolysosome will eventually fuse with the membrane and then release the junk into the environment. Unless it's an antigen-presenting cell. And if it's an antigen-presenting cell, what's it going to do with some of that stuff? Show it on its surface so other immune cells can see it. Yes, Tom?
If it shows the parts of the antigen on its cell surface, won't other white blood cells think it's an emitter?
Yep. Yep, and that's an important part in the activation of those other white blood cells. So these cells actually show it to the white blood cells, and that's an essential step in activation.
But then wouldn't it try to attack the cell that
Actually, it doesn't, and we'll talk about why that's the case later on. We've got to talk about another set of receptors called MHC receptors before you can understand that. That's a very good question, but no, there are mechanisms that the presenting cell has to keep from getting killed as it shows that antigen to the white blood cells. Yep.
I guess, how does the lysosome know which parts to degrade and which not to, and then how does it know what parts to put on the cell surface that are important?
We'll talk about this more later on, but actually, all human cells display on their surfaces, and they do so in conjunction with these molecules we haven't talked about yet called MHC receptors. But pretty much every protein that you produce in your body is eventually degraded, and little pieces of that are being displayed on your cell surface. But then any protein from something that's infected you will be displayed as well. So really, that's kind of part of your cell signature. Your whole cell surface are displaying not only your normal proteins, but then displaying foreign proteins that have infected your cell. Okay, asking some good questions. One quick last thing. What would happen then if a white blood cell burst? Okay, but it might be kind of neutralized by the more basic cytoplasm.
Well, if this bacteria wasn't fully digested, is it possible that it's still...
Yeah, as a matter of fact, there are some bacteria that have evolved mechanisms so they can survive inside the phagolysosome. So if eventually that bacteria has survived and then the white blood cell dies, now the bacteria has been disseminated to a new part of the body. Another thing that can happen is that substances released from these white blood cells when they die, because remember bacterial cells produce the leukocytins as well, is that they can stimulate fever. So some of those molecules that are produced, I'll tell you this term again, but they're called pyrogens, can cause then fever to spike. Okay, so we will talk next time to wrap up about a couple of other things. We will talk about interferon, we'll talk about complement system, and then if we have time, we will talk about inflammation.
Tomorrow will be the last day.
Tomorrow's the last day.
For-
Yes, certainly can.